Hello and welcome to the Sainsbury Welcome Center for Neural Circuits and Behavior for our annual public lecture. We are delighted to have many members of the local community as well as students and researchers from across UCL, and of course I see lots of familiar faces from Sainsbury Welcome Center too, so thank you all for coming. We also have here Esther Rose and Karen Newby, our BSL interpreters, and also Petra Korlevich online, who will be live illustrating the talk today. And those of you on Zoom watching online, you can view the live illustration by switching between the tabs at the top of your screen, and also you will have opportunities to ask questions at the end of the talk by posting your questions in the chat. Okay, so this is our 8th annual lecture at SWC. The purpose of this lecture series is to engage our minds in subjects and make us think more deeply about brains, minds, and behavior, including understanding intelligence from different perspectives, humans, animals, and machines, and also to inspire us about the impact of neuroscience that it can have on society, and I think Stan will touch on that today. Our inaugural lecture was given by Sir David Attenborough back in 2016, who talked about the perception of beauty in birds, and subsequent lectures have covered topics such as animal emotions and empathy, consciousness and free will, the origins of culture and modern human behavior, the psychology of crowds, and most recently, the origins of life and intelligent behavior. So today, we're delighted to host Professor Stanislas Dehaene, who will speak about the neuroscience of reading. Stanislas is a professor of experimental cognitive psychology at Collège de France in Paris, and directs Neurospin, France's advanced neuroimaging center. His research explores some of the most fundamental questions about the human mind. How do our brains learn to read? How do we understand numbers? And what makes the difference between conscious and unconscious experience? His discoveries have transformed our understanding of learning. He's shown that our sense of numbers is deeply connected to how we think about space. He has identified a specific brain region that becomes specialized when children learn to read, a region that originally evolved for other purposes. And perhaps most remarkably, he has identified the neural signatures that distinguish conscious from unconscious experience, what he calls the global neuronal workspace. But Stanislas is not content to keep these insights in the lab. He has been instrumental in translating neuroscience into real-world impact on education. So since 2017, I just heard, he heads France's Scientific Council for Education, where he and a distinguished team of cognitive neuroscientists are shaping how an entire nation teaches its children to read and learn. And I've just learned earlier today that while Stan has been constantly there at the Ministry of Education, there have been 8 ministers that have turned over during this time. Now, importantly, the changes that they have introduced are already showing results in nationwide evaluations, including screening, longitudinal screening from around 3 million children. His contributions have been recognized with extraordinary honors. He's a member of 9 academies, and he's received numerous awards, but including the highest prize in neuroscience, the Brain Prize, and remarkably, the McDonald Centennial Fellowship, a prize that is just awarded once per century. Okay. Stanislas is also a gifted communicator. He's written several books for general audiences, including The Number Sense, Reading the Brain, Consciousness and the Brain, and How We Learn, which have been translated into more than 15 languages. And he's also created television documentaries and authored more than 500 publications. Now, his work sits at the heart of the questions that we grapple here with at SWC. You know, how do neural circuits give rise to complex behavior? How does the brain change with experience? And ultimately, what we can learn about ourselves by understanding the brains. Now, personally, what I think is remarkable about Stan, that even exceptional scientists rarely make genuine real-world impact on society. And I think Stan is unique in standing out in this way, and he should all be an inspiration to us. Thank you. So I'm tremendously excited about what you're about to tell us today. So please join me in welcoming Professor Stan Dehaene.
Thank you, Tom, for all your exaggerations. and especially for bringing me here. It's a great pleasure and a wonderful honor to be giving this annual lecture. And yes, I will be talking specifically about a sort of update of this topic of mine, which has been going on for 25 years in the lab with many colleagues, with my wife Ghislaine, with my colleague Laurent Cohen, trying to understand the circuits for reading and trying to understand how education changes our brain. Before I start, this is the place that I am a scientific director of, Neurospin. Each of these arches has a brain imaging facility, basically. Well, anyway, I just wanted to say that these arches at Neurospin have some of the best instruments to look at the brain, non-invasively, especially fMRI, and we have this incredible beast, Isolt magnet, that arrived already quite some years ago, but it's now starting to give us some results. And you know, we are very excited about the possibility of having higher resolution images of the human brain. This is just anatomy. For the colleagues who visited recently, we are just starting to get fMRI now, and it's beautiful. So it's going to be an exciting time. But at Neurospin, we are very interested also in studying child development. And this is mostly the work of my wife when it goes all the way to babies. We're trying to adapt all of the measurement instruments so that we can study safely young children, including babies, and try to understand how their brains transform when they undergo education, schooling. So yeah, so I'm going to show you some images that we get out. Excellent.
Sorry, Stan.
Out of these, you know, attempts to image the child's brain. Do keep in mind that it's difficult. Children are not the easiest subjects, but we're trying to get the most out of them. And so today I will be speaking about reading. I'm using this Rosetta Stone, which we all know is not far from here. Should have been in Paris, by the way. But anyways, forget about that. So if you had not learned to read, every page of every book would look like would look to you like this. A sort of texture, but no meaning, no understanding. But because you've learned to read, and this is really a miracle of reading, you turn this into speech, into language. And this is what Francisco de Quevedo has called the miracle of reading. We can have a conversation with the dead, a way to listen to the dead with our eyes. Right? And that, of course, is the basis for our culture, because we can store the thoughts and the feelings of the dead, and a lot of knowledge as well. So I would like to describe to you advances in how we understand how we learn to read and why sometimes fail. I will start with a review of the brain mechanisms for reading acquisition, and especially for the scientists here, talk about what we understand now about the neural code for written word recognition, which we think we've made quite some progress there. And this can be used to diagnose some forms of dyslexia. And if I have time, I will talk at the end about what we can do for the Ministry of Education to facilitate the assessment and the acquisition of reading for all children. I'm also not sure why this is blinking, by the way, but that's all right. It always comes back. So what changes in the brain when we learn to read? We have to consider the starting point. The starting point is not a disorganized brain, it's a pretty organized brain. We know that right from the start, spoken language is getting in the brain, the child's brain. This is an image we obtained some years ago already of the child's brain at 2 months. Doing fMRI, and you can see that when the baby's brain is listening to spoken language, it's already the relevant areas of the brain in the left hemisphere, in the left temporal lobe and Broca's area that are already being activated. They also are organized in time. This is the colors here, so that the auditory areas are the fastest, and then we see this chain of activations for higher and higher areas, including Broca's area. What I'm showing you here is that spoken language is, in a certain sense, a natural thing for the human brain. And this is not the case for written language. For written language, we really have a new cultural invention that has to fit into this existing system. So this is how I view reading acquisition. As I have called this the neuronal recycling model. Before the child learns to read, the circuits for spoken language are already way in place, of course, at the age of 4, 5, or 6. Phonology, lexicon, syntax, and meaning have had a long time to be acquired by the child, and the same is true of visual recognition. Faces, objects, spaces, They are all already responsive in the child's brain. And so what we have to do when we learn to read is to recycle, to repurpose some of these circuits. And of course, when we read, the information gets in the visual cortex as any visual information. But what I will show you is that we specialize a particular place in the ventral visual cortex that we've called the visual word form area, or the brain's letterbox as a shorthand. And from there, the information about letter strings gets sent to the language system, and in particular to phonemic representation in the planum temporale. So the grapheme to phoneme conversion, letters to sounds, is one of the key mechanisms of reading acquisition. So we've had data about this for several years now in several ways. First, we did images of the major contrast between people who are literate and people who are illiterate. We went to Brazil and to Portugal to find people who didn't have the chance to go to school. So these are represented on this axis as reading speed. Of course, the illiterates are can read 0 words per minute. You would be at least 100, maybe way above. And we also had some people who had not been to school, but were ex-illiterates in the sense that they had learned some reading in adulthood. And what I'm showing you here is all of the areas of the brain whose activation increases the more you can read. When I present you a short sentence. So what do we see? We see very much what I was showing you on the preceding slide, that language areas of the brain get activated in proportion to how well you can read. Okay. So they're all correlated. Their activation is correlated with how fast and how well you can read. But you also see these major changes in the ventral side of the brain. This is the left hemisphere seen from the bottom. And you see what we call the visual word form area. It's actually an extended zone of activation where we see activation in proportion to how well you can read, a reorientation of visual resources for letters. And in the same study, we had the surprise also to find that if I present to you spoken language, whether it's sentences or words or pseudo-words, the activation for spoken language can be almost doubled in people who have learned to read. So also on that side, in the superior temporal region, there is a change of activation when you learn to read. We think that this is phonological awareness, the ability to hear and distinguish better the phonemes of language. And we also found anatomically that the connections between these two areas was reinforced, suggesting that this is really a key circuit that gets changed when you acquire reading. An interesting aspect of this study is that we could ask what would happen to the visual word form area if you had not learned to read. In this study, we had all of these visual stimuli, including letter strings. We could replicate that when you present letter string, there is a strong increase in activation when you are a better reader. We can measure about 50% of the variance in reading scores, in reading speed, by measuring the activation of that region to written strings. So that's this region that you can see here in the left hemisphere being nicely correlated with literacy when you present letter strings. But what does it do with the other categories? We found that it can be very strongly activated, for instance, by faces, also by tools, and to some extent by checkers, and that many of these activations decrease with literacy, suggesting that there is a sort of competition, and it makes sense, right? The number of neurons is essentially fixed at birth. We have some cortical space. Well, if it is taken by reading, it's not going to be taken by these other categories. There is a sort of competition. And we did find, and this has been a repeating finding in several studies, that the better a reader you are, the more your face recognition is shifted to the right hemisphere, suggesting that there is really this sort of competition for space. If you acquire reading, and in 96% of right-handers going to be in the left hemisphere because you have left hemispheric language, then the face system is going to be will have to be shifted a little bit more to the right hemisphere. This is all in adults, and it's a huge comparison, literate versus illiterate. So then we went to children, and with my wife, we scanned some children in some simpler paradigms where you can show words, faces, houses, checkerboards. You can see what it looks like in a 9-year-old. This is the group of 9-year-olds. You can see a very strong activation to words in the left hemisphere. That's the visual word form area. A tiny little activation to faces next to it. Most of the activation to faces is in the right hemisphere, okay? And also there are responses to houses and checkerboards. Now you can see that this is not the case in a 6-year-old non-reader, and it's not just a matter of age, because if you scan 6-year-old who are already reading, you can see that the visual word form area is already appearing here, and the face system is being slightly reorganized as well. We recently completed the study with more children, and that has been interesting because it allowed us really to better separate age versus reading acquisition. The two are very correlated, especially in France, because when you are 6, you enter school, and the first year of school, you learn to read. That's really the curriculum. But still, you can find children that have learned to read a little bit earlier, so we call them beginning readers. And you see that already you see the appearance of this visual word form area in the left hemisphere. We showed that essentially the appearance of the visual word form area here is dependent on reading scores, on the capacity to read, your fluency, not age. So in this multiple regression, age has no effect. It's the reading scores that drives the activation of this left hemispheric area. Completely different for faces. Faces, and that's a little bit of a surprise, they are slow to develop. They develop all the way into adolescence. And this has been shown by other groups as well. So the response to faces depend on age, presumably because you have to meet a lot of different people, different faces, to tune your face system progressively. And although it starts in infancy, it's a slow process. And that's the way to see what we do when we acquire reading. We force that plastic system to concentrate very much on the strings of letters. In the first grade, we tell the child, you know, that's what your visual system should do. It should be tuning to letters. And we use this plasticity to reorient part of the system towards strings of letters, most cases successfully. What I was showing you so far is cross-sectional, but we can also do longitudinal studies. We were very lucky some years ago to have the permission to scan 10 children many times, every 2 months essentially, while they were learning to read. We had 2 scans before school, and then we had 4 scans all along the first school year, and 1 more scan for 8 of them 1 year later. And that allowed us really to look at single children, how do they change when they acquire reading. And you can see how they change behaviorally. They all acquire grapheme-phoneme relationships. This is the onset of schooling, 0 here. And we chose children that didn't know much before, but suddenly schooling was doing its job and they acquired grapheme-phoneme relationships. Each color is 1 child. And they also acquired slower some fluency in reading. They became more fluent. So what happens in the brain? Very much what I told you now occurring in a single child's brain. This is one of these children. You see very few responses to words compared to other categories of visual stimuli before school starts, and then immediately after school starts, you can see these responses. I can zoom in so you can see better these responses in the sector of the visual word form area, and also in this more superior temporal sulcus region here. And these responses are already at the right place. They are getting stronger as a function of fluency of the child, but they appear very, very quickly as soon as you engage that circuit for grapheme-phoneme correspondences. Compare that to the face system. The face system is already detectable in the run 1 or run 2, and it's very hard to see that it's changing. It's changing a little bit during this time period, and it's slightly correlated with reading scores, but essentially it's changing much more slowly. One cool thing about that study is you can look backwards in time. We could take the scans from the last sessions of these children where they have a visual word form area, they have specialized the visual area for recognizing letters, and then we can travel backwards in time and ask, what was it doing before? But now really in a single child. And we saw several things here. We had several surprises. One surprise is that you already see the onset of these words responses. So these, sorry, oh, this is going too fast. These are the voxels that are going to specialize for words. And if we travel backward in time, we saw that they were not responsive for words at the beginning, but actually they were responses to objects, not so much to faces. You can see actually faces are quite low on this graph here. But they were responding to objects, and they keep their preference for objects for the most part. They're also responding a little bit to numbers as a function of time, but mostly they are appearing in this particular region, quite different from all of the other categories. If I find voxels that respond to faces in a 7-year-old, They were already responding to faces 1 year earlier, but not that for words, right? So for words, we really create a new category, and we create it mostly out of voxels that have a slight, quite moderate actually, preference for objects, and not so much for faces. So how can we understand these results? Just to give you a sort of crisp picture of what's occurring in a child's brain, we think that before schooling, we can already see preferences for objects, for tools, for faces, for houses. There is a topography. We all have in the brain this sort of mosaic of areas, which is in the same order in all of us. But in the child's brain, there are still many areas that are plastic, many cortical columns that are plastic. They are weakly specialized or not specialized at all. And if you learn to read, you invade a specific place in this ventral visual cortex by written words. You tune this region to written words. If you did not do that, so if you look at a voxel, in fMRI we already have a relatively coarse view of the brain, so we don't see single columns or single neurons. We see a voxel of maybe a millimeter size or a bit more. So we are going to see some preference for words among preferences for objects, right? But if you had not learned to read, the system would reorient. It would not stay there sitting, waiting for you to learn to read. It's designed by evolution. I shouldn't use the word design. It's evolved to recognize faces and objects. And so the same voxel, if you stayed illiterate and didn't go to school, would tune to objects and to faces in particular. That's the competition process we're talking about. We now make more images of this ventral visual system, and with modern magnets like a 7 Tesla magnet, you can really achieve quite high resolution in individual subjects. So this is a single subject study that we did just a few years ago on bilingual subjects, and this is a bilingual brain. It's just a single subject responding to words in French and words in English, as well as pseudo words. This could be a French word, and this could be, I suppose, an English word. I hope it's not. I think you will agree that these cannot be English or French words, right? They're crazy strings of letters. Maybe they could be Polish, but I don't know Polish. Okay. So what we found here, first thing, you zoom in the visual word form area, it's not an area anymore. It's like when you zoom in with a telescope, and you thought it was a star, and it's a galaxy. It's really like that. We thought this was one area, and now we're discovering these little patches of cortex, very much like for faces in Doris Tsau's work. So each of these patches of cortex is very, very specialized, can be responsive just to words. But it's, I'm sorry, I cannot easily point. You can see that at the input here, in early visual cortex, it makes no difference whatsoever whether the stimuli are in French or in English or in this pseudo-Polish. But as soon as you enter this visual word form system, the main characteristic of this system is to have learned the statistics of the language that you are able to read. So it has learned that certain combinations of letters, certain letters at certain positions, make a plausible French word, and others do not. And it's very sensitive to that, so this is the gradient that you can see here. So we have this idea that this system is tuning in to the statistics of the language, tuning into an alphabet. If you had not learned the alphabet, but you had learned Chinese, it would tune to Chinese. If you have not learned Hebrew, it does not respond to Hebrew. It's really tuning in to specific statistics of your language. So this is a summary of part one. We all of you in this room, if you came to my lab in 3 minutes, I could show you your visual word form area. It's really very salient area that responds to words more than to any other categories. You can see it here actually. It can respond to letter strings more than to anything else, including numbers. Right? Numbers and letters are very similar, but somehow the brain is sorting these categories because they have different functions in our culture. So we know many, many things about this area. We know that if you have a lesion there, you would lose the ability to read. We know that it's operating unconsciously after automatization, can run completely without awareness, even if you're not aware that there was any word, it was just flashed subliminally. And we know, and this will be important for the rest of my talk, that it encodes letter order. It's sensitive to the order in which the letters form a word, which is very important to discriminate anagrams, like form and from. And so that's the second part of what I would like to tell you today. We're beginning to understand what is the neural code inside this area. How do single neurons care and tune in to discriminate different kinds of words. The idea came first from simulations. We had this idea that you could recycle, when you learn to read, you recycle your visual areas. Well, we thought maybe we could recycle an existing model of visual recognition. So we started with this model called Cornet-Z. There are several models that have a high brain score that are supposed to capture quite well the early stages, at least, of visual recognition. They've been designed to mimic a little bit the hierarchy of visual areas from V1 to V2, V4, IT, and to recognize pictures and categorize them. Here, this model has been trained with ImageNet, which categorizes 1,000 types of images. We just took the existing network and we recycled it to become literate. We trained it with 1,000 more categories for 1,000 words, regardless of size, font, location on the screen. And we looked at what was happening in this model when it became literate, and we found that there were a number of units, 33 in this particular instance, but more like 60 sometimes, that looked very much like the visual word form area. They would respond just to letter strings, not to other categories of visual images. And then we found that if you looked at these units, there was a sort of barcode. Each word had a separate set of active and inactive units that created a sort of barcode that was specific to the particular word. So you can see here words that are quite similar with each other, but they differ slightly in one or two of these bars, which is enough to say that this is a specific word. So we thought we could understand that and then go back to the real brain. So let's look first at what this model is doing. We modeled the units of this model by regression, by presenting it with a lot of words with different letters. So there are 26 possible letters from top to bottom here, and there are 8 different locations for these letters inside a word. We played with several location coding schemes, and we found this is the best explanation of what the units do. They care about letters from the left and from the right of the words, and so you can find each of these panels is a different unit. You can find units that have tuned to letter S, or to the letter U, or to the letter A, and you can see in this graph that this is a unit that is tuned to letter A, but only at a specific location, okay? Only on the left of the word, maybe location 1, a little bit location 2, right? And it has done so regardless of whether it's uppercase or lowercase. This is a unit that is tuned to letter R and has become tuned to that only in the position before the last, okay? So when you analyze all of these units, you begin to understand what they are doing. They have tuned to letters, but many of them have also been tuned to a specific ordinal position inside the word. They tune to letter A in the first position. And you have many units that care about the first position, some units that care about the second or the third, some units that care about the third from the last, or second from the last, or the last, okay? And the tuning is very much like in number neurons. It's very precise for the first and the last. It becomes less and less precise as you go inside the word, right? And that creates some fuzziness inside the word. We also understood how the units become tuned. There is a very nice computational problem here. The input is on the retina. The coordinates are retinotopic. But if we are correct, these units are not retinotopic. They care about ordinality. They care about first, second, regardless of where the word is and how large it is. So how is this done? How is this transition? Well, we thought we could understand it by presenting this sort of stimuli where you can see that there is one letter, O, which might be the preferred letter for a unit, and we vary its position we vary the absolute word position, and we also vary the relative position of the letter within the word. So if you think of how units might fire to this sort of stimuli, well, there might be some retinotopic units that would care only about whether there is an O in the receptive field, and because we vary its location, these units would have this sort of diagonal pattern. They would really like the letter O to stay where it is in retinotopic coordinates when you move the words, so they would have these diagonal patterns. But if you have an ordinal unit, it should really stick to responding to the last letter O, regardless of where it is, actually on the retina in front of the subject. So we think that the brain is moving from this at the input, retinotopic letter position, to this ordinal letter position. And can we understand how it works? We figured it out finally, and I'm very happy to report that this was a recent publication by Akash Agrawal, who was a postdoc in the lab. So let me start with something simple. There are these units at the output that care about a single letter anywhere on the word, inside the word, anywhere on the screen. That's very easy to understand because that's exactly what these networks have been designed for. They are convolutional neural networks. So they start with a very precise receptive field. You find units that are tuned to a particular letter right from the start, but they are tuned to a specific retinotopic position. But because the model is convolutional, these receptive fields get duplicated at many, many positions, and they get broader and broader because of the connectivity from V1 to V2 to V4. And as they get broader and broader and broader, they eventually get location invariant. That's exactly what these networks have been designed for, to get location invariant recognition of specific objects, such as a letter S. Okay. So that part, I hope, is easy to understand. If you're vaguely familiar with these networks, that's quite easy. But what happens with these units that have ordinal coding? They care about a specific letter, but only if it's the first or the second of a word. How could they do that? Well, we analyzed all of the layers, and we found that there are no such units in V1, but in V2, you can find a unit like that, which cares about a specific retinotopic position, but also about a specific ordinal position. And that was a little bit of a mystery. How could you do both? Okay. But we figured out it's very simple. This unit cares about two things. It cares about letter A, for sure, but it also cares that there is no letter left of it. It cares that there is a space. And that's a normal receptive field. That's an easy receptive field. It just says there should be a space and letter A. It gets inhibited if there is another letter left of it. Right? And we could trace the inhibition through the circuit. So as soon as you have letters like that, units like that, if you make the receptive fields broader, and that's exactly what these networks are designed for, you get broader and broader tuning, but still high sensitivity to the first letter, okay, or first and second. And it gets broader and broader in the hierarchy, and when you reach the higher layers of the system, you get these purely ordinal units. They no longer care about a specific location, but they care that it is an ordinal position inside the word, that there is a space left of them, or right of them, or quite far left of them. So you get all of the ordinal position tuning that we could see in just understanding how these units respond. So we call these space bigrams. And it's a new idea. Several people had this idea that the brain should be caring about bigrams, pairs of letters, E left of N, something like that. That would be a very good code for words. Well, that's not exactly what we are finding. There are a few units that are like bigrams for letters. But here, the bigram is a letter relative to a space. Okay. And with certain position tuning of the two space versus letters. And it's variant of the same concept, I would say, and we should have thought about it because, well, do you guys know what is the most frequent character in English or in French? E. Everybody says E. And I would have said E, but actually it's not true. The most frequent character is space. So it makes sense that the brain is tuning to space plus some other letter, and it's also tuning to some of the frequent bigrams, but not so frequently. Okay, I said the brain, but remember, we're still in the model here, where we can analyze every synapse and understand every synapse of the model. So the question is, will this transpose to the real brain? The prediction is very simple. There should be a period of time where you are dominated by retinotopy, and you might have retinotopic letter detectors, but only late in the system would you have ordinal coding, where the system really cares about letter 1, 2, 3, 4, etc. So testing this is quite hard with current brain imaging methods, but we did manage to test it with the same exact idea that we had for testing the network. So first of all, you need high-resolution images. We have 1.2 millimeter, 7 Tesla. It's not so high, but it's okay. And the second idea is that we're going to put one frequent letter, such as letter O, and we are going to surround it by infrequent letters. So the logic is that the frequent letter will be highly represented. It should create firing. The infrequent letter should essentially not perturb too much the activation, but it should give a frame of reference for the position of the frequent letter. It should say the The frequent letter is first, middle, or last. Okay. So we created this space of stimuli, 12 stimuli here, times 3, because we had 3 letter identities, T or E were also tested, and we presented all of these stimuli to subjects and fitted their neural responses with the same exact model that we fitted to the network. So this is still the network model here. You can see that across the successive layers of the model, word position decreases, retinotopic letter position increases, specifically when you've learned to read. So the red bars are for the literate network. Illiterate networks have some of these units, but few. And especially ordinal letter position massively appears in the last layers of this network where it's been trained to become literate. So these are the predictions we started with, and it worked very beautifully, just appeared a few weeks ago in PNAS. If we do fMRI and we concentrate on a few regions of interest, because these are really subtle effects, you can see that the word position effect decreases from early visual areas to the visual word form area in lateral occipital cortex. There is tuning to specific letters at a given retinotopic position, but above all, there is a significant additional contribution of ordinal letter position. So we think that in order to account for the brain signals, we have to assume that the ordinality of the letter, regardless of where the string appears on the screen, is encoded. And the same with MEG signals. So this is magnetoencephalography. Now we have a time course here. 0 is the onset of the flashing of this string. You can see that, of course, word position, this huge retinotopic component, dominates the signal. But a little bit later, we see the appearance in proper order of the retinotopic letter code and then of the ordinal letter code, right? And if we try to reconstruct the sources of that, it's never very precise, but we have an early occipital effect and we have a late lateralized ventral, primarily ventral effect here. I wouldn't, trust too much these sources for MEG, but they are compatible with this idea. So we think the brain transitions from retinotopic coding, well known, to something that was not known before, these space diagrams with ordinal tuning. Of course, it'd be nice to have human single cells, and I turn to one of my favorite neurosurgeons, Itzhak Fried, who started the possibility of recording from human single cells, and it's still quite some endeavor, I have to say, but in some epilepsy patients, possible to record from single units in the human brain. And lo and behold, with my colleagues Nama Friedman and Yair Lacrest, we had very, very few electrodes inside the fusiform gyrus, because it's not a very frequent site for these recordings. But we had a number of cells, you can see 2 here, that were tuned to specific letters at specific positions inside the words. This unit likes many angular letters, but they have to be on the left side or in the middle side. It doesn't respond when the same letters are presented on the right side of the word. And this one was particularly nice. It really likes W, but it has to be on the middle or on the right of the word. And these are all, by the way, contralateral to the recording side, so it makes sense. But they are really ordinal coding rather than retinotopic coding. They are very anecdotal, so that's not, very strong scientific evidence. This is still unpublished. But more interesting, I think we can ask these questions. Could you get data from monkeys? Now that sounds crazy, right? Monkeys are not going to learn to read, and they don't, I reassure you. But they can recognize visual objects, so why not letters? They can be trained to recognize letters. And furthermore, if I'm right with this recycling idea, even before they are trained, they should already have some tuning to those shapes. Those shapes are useful to recognize the environment. So we could discuss more about that. But a T-shape, there are plenty of T-shapes in your environment. When one object hides another, there is a T-shape at the junction, right? So we think that many of these shapes can already be present in a monkey. So it's not completely crazy. So we collaborated with SP Aron in Bangalore, has this really interesting lab where it's completely wireless, and the monkeys come when they want and put their chin on the chin rest and start the next trial. And we exposed them to a system of words, 36 words that were made just of these 3 vowels and 3 consonants, and creating sort of pseudo words like this. They are pseudo words for us, but they were words for them. Beware, there is only orthography, there is only spelling, there is no phonology, there is no semantics. It's just stimulating this sort of learning of the statistics of a visual stimulus. So we trained the monkeys for 5 days to do the same different task, and we measured the tuning before and after they were trained. And maybe I should go a little bit fast because it's complicated and not all of you are, passionate neuroscientists. But one monkey clearly was able to learn, the other was not able to learn, but both monkeys performed above chance. Both monkeys had the capacity to decide that two words are different when one is made of completely unfamiliar letters. So this is the example here. You get a sample word, and you get something which is made of completely different letters. Monkeys could reject that as non-match, and vice versa. And even more interestingly, both monkeys could use the number of letters at a given retinotopic position, but also at an ordinal position, to facilitate the same response. So they had some sensitivity to the ordinality of the stimulus, and this was getting better in the monkey 1, not in monkey 2. And then we recorded from them, and applying the same exact model as before, we could see that there are units that are tuned to letters in the monkey, in the inferotemporal cortex. You see here all of the word stimuli, 36 times 5 possible positions, and this unit likes a few of them. These are the top. They all have an O in a certain approximate retinotopic position. The model says this is a retinotopic unit that likes the letter O at a specific retinotopic location, just contralateral to the implantation of the monkey. But here's a more interesting unit. Again, there's some firing, and then it becomes selective to some units, to some words. Look at all the words that it prefers. They all start with O. And now it's relative position tuning. It's ordinal. And the model fits, says, this unit likes letter O, but it has to be in the first position, okay, next to a space. Okay. So exactly the sort of units that we saw in the model and that we predicted should be these space diagrams. So there's a lot of work on these monkeys. I'll spare you all of the details, but maybe interestingly, we can see that the ordinal code is already present before training, but it gets refined, and there is more ordinality in the firing of the cells after training with orthography compared to before training with orthography, just in the monkey that was able to learn this position invariant recognition of words. The other monkey who didn't learn had a refinement of the word position and the retinotopic code, but not of the ordinality code. Okay. So this is all consistent with this idea of refining an ordinal code as you learn to read. We think we have a handle on the neural code for reading, still an approximate handle. That's the reminder of the sort of model, space diagrams. In the last 5 minutes, maybe, 5, 10 maybe, I want to tell you a little bit about how this model can illuminate some of the findings in dyslexia research. What could go wrong with this model? First, if we just look, we do the same exact experiments as we were doing before with children, but some of them were not able to learn to read very well. So we scanned 9-year-olds, and I already showed you the readers, but now we have children who are struggling with reading acquisition at the same age. You see two things. You see a lack of the visual word form area. That's a very strong biomarker of not learning to read well. And we also see a disorganization of the visual system. And this was significant for faces. So it's quite interesting that the dyslexic children that we scanned had lower activation in the left hemisphere for words and lower activation in the right hemisphere for faces. Again, suggesting that there is this sort of competition. The visual system gets reorganized, and it doesn't get reorganized in a proper way if you are dyslexic. Now, beware. We see a correlate, but that's not necessarily the cause, right, of dyslexia. They are just not learning to read, and this system is not tuning. And in fact, if we look at the whole brain, we see that there are not just the visual word form area, but a number of areas that are different in dyslexics. Interestingly, there is a cultural universalism here. So many of these experiments, by the way, we've done also in China or in Chinese readers, and we find a lot of commonality. The circuit for reading is very much the same. And as you can see here, the lack of visual word form activation is very much present in both French dyslexics and Chinese dyslexics who are also struggling to learn to read. So that's a very general marker here, and it's present in individual subjects. It's not just a matter of more variability in the group of dyslexics or something like that. It's really a lower activation peak. But it's a circuit. The whole circuit is to some extent hypoactivated in dyslexic children. They are not reading, and that circuit is not engaging as it should when you learn to read. So what could be happening? Well, I think it makes sense to me not to speak of dyslexia in the singular, but to speak of dyslexias in the plural, because it's a whole circuit that you have to tune in order to learn to read. I showed you the major components of it at the beginning, and so it would make sense to think that any of these successive stages could cause deficits in learning to read. And people in the past have insisted a lot on phonological deficits as a main cause of dyslexia. But if this view is correct, this connection from vision to phonemic representation could also be a cause, and the impaired orthographic code itself would cause a major impairment in the ability to learn to read, right? So we began to look for the possibility that there might be these other forms of dyslexia. And Professor Nama Friedman was a pioneer in this. She's been doing that for 25 years and has discovered that there are, in fact, very different types of dyslexias. What could be the predictions from the model? Suppose we look at the neural code for badge. So you are looking at the word badge, and right now in your head there should be units that care about B, right of a space, A somewhere right of a space, D and G somewhere in the word, G left of a space, E left of a space. Okay, that's the neural code for badge. Well, notice that the neural code would be very, very similar for bagged. Okay, and in fact, if I just show you this, you might say it's just the word badge, no problem. So errors in reading may occur because you don't have a precise enough code, especially in the middle letters, which have this coarse tuning for ordinal position. And we all know that exists because you've all seen this email that tells you to read something about Cambridge University, but it's actually Cambridge, right? Cambridge University. We can read words where there are internal transpositions, and you get priming effects from transpositions of letters inside words. But it also is the case that there are children that struggle specifically from this aspect, and they read form as from. They switch letters internally within the word. Okay. Now, another thing that we might predict from the same neural code is what would happen if you process two words at the same time. So suppose I showed you the words fun badge, and that would be the neural code for fun badge. Okay. Well, the problem is that the same exact neural code for fudge ban. Okay. And you wouldn't know which one it is. Well, it happens in normal subjects. If I flash you two words and I don't leave you enough time to sequentially look at them, you're going to make illusory conjunctions of exactly this kind, mixing two words. And also, there is a form of attentional dyslexia. It's been reported with adult cases by Chalice and Warrington a while ago, and it's been reported in developmental cases. And these children will do exactly this sort of errors. You give them win-fed, and they might say fin-wed. They don't know which letters belong to which word. Notice how different it is from the previous case, right? Because now the position is correct. The ordinal position is completely correct. The letter F is coming from the first position, but it's just assigned to the wrong word, right? So completely different patterns, right? So we looked for that in French children. This is a recent publication. I think I have to go a little bit fast here and skip the details, but I think there are two simple but powerful ideas for dyslexia researchers because this is still controversial in the field. One first idea is that you should monitor the error types and not just the error rates. There is a lot of research that just says you're dyslexic because you make 15% of errors in a standardized test, and it stops there. You have to look at the types of errors to distinguish this. And you have to select target words that can be misread as other words. You have to select stimuli that are likely to cause this sort of errors. Otherwise, the children may not make this sort of errors very apparent because they can correct from the context and so on. So we created this long list. We call it the Malaby screener, after the delicious dessert that we were having together in Tel Aviv some years ago when we were designing this test. Try the Malaby if you have not tried, it's very good. So we have this list of words, pseudo-words, word pairs that are especially designed so that you can make a lot of these internal inversion errors, letter position errors. These are all French words, but I think you get the idea. Cygne versus singe, just a letter transposition. We can do that with pseudo words. And we also have these word pairs on the right that allow you to swap the letter of one word with the letter of another word and create these attentional dyslexia patterns. We found double dissociations between children of one type and of the other. You can really have just purely selective letter position dyslexia or purely selective attentional dyslexias. And these are actually quite frequent compared to the other types of phonological deficits or surface regularizations. They can be confirmed by a second test, so there is no double dipping in this data. And I don't have time for the details, but there's one thing I want to say. These children were missed. They were all in a special school. They were all complaining about reading, and they were assigned by their parents to this special school, but the classical dyslexia tests were not picking them up, and they have completely normal phonological awareness. So phonological awareness is not always the criterion, and I think from all we know about the neuroscience of reading, the development of this visual word form area is a key step that could go wrong in some of these children and could create some of these deficits. One word about rehabilitation. Once you have a good diagnosis, you're much more likely to rehabilitate these children properly. And it's not going to be more grapheme-phoneme learning. They know their grapheme-phoneme correspondences, right? It's not the source of their problem. So how could you help them? Do you have an idea of how you could help them? You could just space the letters for the first type, for the letter position dyslexia. This would help to space so that the position is more clear in the word. And you could also help them by tracing with the finger. And Nama Friedman has shown that they are really helped. If you tell them just trace the word with your finger, they can develop better reading. What about the attentional dyslexics? Those that mix form pair and farm pole? What could you do? Very simple idea. You could cut a piece of cardboard and make sure that they see only one word at a time and slide it over the text. And suddenly reading becomes normal. So it's not grapheme phoneme or whatever, it's really something very particular about the difficulty of selecting one word, paying attention to a single word inside the reading system. So I think we're just starting this line of research in France, but it's been going on in Nama Friedman's lab for a while now. It's really, really important to distinguish these subtypes of dyslexics. To some extent, it also kills what I was telling you before, because we're really in brain imaging. We shouldn't pool all of the dyslexics together. If we pull them together, probably we just see the consequence, the common core consequence of being dyslexic. But in order to understand the system better, we really have to make these distinctions between different types of patients. Voila. Tom was kind enough to tell you that we are trying to make this applicable to French children in France. There is a scientific council for the national education system. I'm the president for 8 years now. It's been quite exciting the date is wrong, I'm sorry. It's 2018, I think you were right. We create documents for teachers, for teacher trainers, in order to tell them about the science of reading and especially the importance of identifying the graphemes, mapping them to phoneme. There is a beautiful convergence between neuroscience and educational sciences, which have all shown that the best way to acquire reading is to have phonics, synthetic phonics, where you have explicit teaching of how letters work, how the string of letters work from left to right, and how you can convert every letter or group of letters into sounds. Okay. We have created this evaluation system, which is really nice. Every kid in France in the first grade gets evaluated at the start of the year, in the middle of the year, and at the beginning of the next year. Okay. And so we have these incredible statistics about all of the children of France in this longitudinal manner. And it helps. The main goal is not science. The main goal is to help the teacher notice what the kids need. It's very similar to the phonics check that we have here. This was a source of inspiration. It's a bit broader in cognitive outlook, so we also look at mathematics, and we have very interesting statistics on mathematics as well. But it also helps us to look at what works. And for instance, in a very recent study, we were able to show that even though there is no longer whole language methods in France, there is still balanced reading, and there is still sight words. People have to memorize sight words. And we found that those teachers that are using sight words and balanced reading, they do worse than those that just focus on synthetic phonics. We can see that in the national statistics. So we can make recommendations from teachers about what is the best way to create this transformation in the brain. We can measure fluency. I don't think we should underestimate, by the way, the power of cognitive science to measure things properly in the schools. It's a very useful contribution of our field. And so we're measuring fluency. We're telling teachers that this is in 6th grade now. We're telling them that there are specific children with special needs because in 6th grades they are not at grade level for reading. We can measure the reading speed. We like that teachers attend to the kid and actually measure their fluency on a one-to-one basis, but it does take 1 minute of a human being to attend to a child and measure the fluency. But we can also measure it on the computer. In this case, we just ask children to do lexical decision. They see a word or a pseudo-word, and they have to say, is it a real word or is it a pseudo-word? And you can see that we can measure the speed with which they process words of different numbers of letters. And that's a very good proxy for fluency. If you are a struggling reader, you are still a letter-by-letter reader. So you still have this huge length effect. You see the reaction time is increasing with the number of letters in a word. Now, all of you in this room, you are more like here. You have a flat curve where words that are short and words that are long, they are all processed in parallel across the visual field, just like in these neural networks. So we can measure that, and we can detect whether children are struggling with specific types of pseudo-words, with specific confusions. So we begin to have diagnostic tools that are being spread throughout France now. And we also use these tools to debug snake hole. I'm so sorry to say this. In the field of dyslexia and reading research, well, first of all, I receive a reading method every week. All right. So please don't send me your reading method. It's all about colors, moving, I don't know. The imagination is without bounds. But the same for dyslexia. And people claim things like you should wear glasses for dyslexia. And of course, it's crazy. It's not at the eye level, right? So in France, we had to make this publication to fight a major French glass company that was selling glasses and also lamps that are blinking. And supposedly the blinking, which is very fast, is helping you to read. And we are showing it's not the case at all. With these very fine measurements, of course, dyslexics are not being helped. If anything, there is a little bit of a placebo effect, of course. So if we tell some of these people the lamp is on, they get a little bit better. But that's not an effect of the real lamp. Okay. And I just finish with this because you might be interested. The lab has created a non-profit, which is called Excello. And we've created this reading method called Kalulu, which is based on a lot of things. Teacher manuals, booklets for every child, multiplayer collaborative games, and tablet software in the end to facilitate reading acquisition. It's a very systematic synthetic phonics method. Synthetic phonics comes in different grades, and here we've tried to really squeeze in every single thing we know about the science of reading from our lab, from other labs, to facilitate the engagement of the child and to facilitate the speed with which this transformation is occurring in the child's brain. So this is Kalulu. We're deploying it a lot in Latin America, in Spanish or in Portuguese from Brazil, because there is a lot of demand there. There's a lot of still inappropriate teaching based on the whole language method, and we know this is not the right way to go. So this is Kalulu invading the planet. And with that, I will stop there, and thank you very much for your attention.
Stan, thank you for a really inspiring lecture.
Thank you.
We have time for some questions from neuroscience to societal impact.
Thank you for your talk. What happens in humans with the, do we see any plasticity similar to what we see in child?
In human adults, you mean? Yes. Okay, that's an interesting topic which is not sufficiently studied. The plasticity should be more limited, and that's what we think we see. First of all, it's quite difficult for adults to acquire reading if they have not been to school. It's also very difficult for us to learn a new script if you have not learned it early on. And we scanned some illiterate subjects while they were learning. This is still going on in collaboration with Brazil. The changes are very slow to occur. They do occur in the visual word form area, probably because this region has the right connectivity to the language system, but they also seem to be much broader in illiterate people who learn to read. The activation is not focused in these very small patches of cortex as it is in readers that have learned from school, right? So presumably, yes, the plasticity is closing, and that would make sense relative to all we know about the sensitive periods for acquisition of vision.
We have a question from Claire Mooney online. If the child learns to read late, can the voxels or areas of brain that may have developed be used for faces or tools instead of reading be repurposed to support reading functions? So kind of a delay.
Yes, I guess that would be the prediction. I don't think we have such cases. At least in France, the schooling is extremely well regimented, and you are 6, you have to go to school and you have to learn to read. There are countries that delay it by 1 year, but not much more than that. So I don't think the case is known. I also think that the window of plasticity is quite large. If it closes, it is relatively late in this ventral visual system, unlike the early visual system. So presumably you could still learn to read quite fluently if you learn at the age of 8 or 9 or 10. We don't really know this directly, but I would not be too worried about having to learn at a very early time point. On the other side, it's also interesting. Could you learn to read earlier? There are many instances in families, informally, that suggest that it can happen. What I think is the critical variable for reading readiness is really the state of your spoken language. The visual system is always ready, but the spoken language system has to have a critical mass of words and a nice lexicon of spoken language before it's meaningful to begin to learn to read.
I'm really interested in how the translation of the use of the fovea to read, so neurons in the retina that have very small receptor fields, subtend the angle of a letter, right?
Right.
Then translates into neurons in higher visual areas that recognize specific letters just as part of their own receptor field property. Do we have an understanding of that? And is it kind of connected to that when we learn letters? Normally we're presented with things that are much larger, right?
Yes.
To see, and then as we progress through learning to read, we start to be able to recognize letters as small text.
Right. So you're completely right. I skipped over that, but even in the illiterate study and in several other studies, we found changes in early visual cortex as well. And now I think we understand better because even the retinotopic cortex has to tune in to the shapes of letters to facilitate this whole pyramid that ends up with the visual word form area. So we do see that in a few studies. It's not very well studied, and I agree with you. The prediction should be that perhaps there is a sort of backward going process where you start at the top. There is this theory, by the way, called reverse hierarchy theory, which I think would fit the bill. So you would start at the top, and then as the system becomes better and better tuned, you could specialize earlier visual cortex, and ending up with what we see is a little bit of specialization inside even early visual cortex, perhaps all the way to V1. Right? And yes, as you do that, you could learn to recognize smaller and smaller print, which is exactly what is occurring in schools. We go to smaller and smaller print. It's probably also dependent on eye movements, and this is really a blind spot in everything that I've been telling you here. We are also training our eye movement system when we learn to read, and as we get more and more precise, presumably we have less and less need for spatial invariance because the eye is fixated near the center of the word, and retinotopy can then help. So all of these processes are probably going on at the same time, and yeah, that requires more study. We've never studied eye movements in the lab. I feel it's a sort of a dark spot in what we did.
So a question here?
Yeah, so a question I've been interested in for the last couple of years that I haven't seen a great answer to, and I was wondering if you had insight onto it, was why, if the circuits are relatively, basically, why is there specialization to the left of the left of the hemisphere for the visual word form area?
Yeah.
If the circuits are more or less the same across the hemispheres, and it is a competition type of thing that's happening, why don't we see equal parts people with visual word form area in the right and to the left.
Right.
And then what's the nature of the connection that pushes then the facial recognition to the right?
Yeah, so that's an easy one. It's the language. Most people have left hemispheric language, even in left-handers, by the way, still 2/3 have left hemispheric language. And we've studied this, and there is a collateralization of the visual word form area to the language system. So it's presumably it's more efficient. The connections are shorter and more direct from left hemisphere vision to left hemispheric language areas. We think, by the way, this particular spot is selected out of all of the ventral visual cortex. This particular spot is already connected to the language system in children, even in infants. We can see functional, so-called functional connectivity, functional correlation with this particular spot of the brain. So it's all about minimizing the cost of propagating the information. Typically, a word starts on the left and on the right of your fixation because you're fixating near the center of the word. So both hemispheres work to recognize the specific letters, but more and more the information gets channeled to the left hemisphere, and eventually it's just taking place in the left hemisphere. And in terms of lesion, you can lose reading essentially only if you have a left hemispheric ventral visual lesion.
Thank you for the inspiring lecture.
Thank you.
You alluded to the focus of the eye on the words, the middle of the word. Is there any difference from learning or reading more from a book rather than a tablet or a smartphone?
Very good question. I used to think that it was the same, and to a large degree it's the same at the input because we make the page on the tablet look very similar to the page on paper, right? Even down right to the font and so on. However, there is a little bit of research suggesting that even though the input looks so similar, we are worse on tablets. And that's pretty well demonstrated now. And there seem to be several reasons. First, we are lost in the text. There is no information that you get from the space of the book about where you are. And it's very interesting. So, for instance, if you ask people questions about the meaning of the story that they've just finished, they know a lot about the characters and so on, but they are specifically lost about the order of things. The order of the events in a novel is lost when you read, is more lost when you read in a tablet. And the other thing which is happening, which is very relevant for education, is that there seems to be this expectation, as you read from a tablet, that you're going to be distracted. And the brain is less focused. And even in cases which can be done in the laboratory where no distraction whatsoever occurs, you're reading the same exact thing for 10 minutes on paper or on a tablet. On a tablet, processing is less deep because it seems that you are less well engaged with the reading because you expect that you could be distracted at any time. So that's pretty bad for tablets, and I think it suggests that we should stick with paper for a little while at least. I'm sure we will find ways in which the interface is better designed so that we have some notion of the thickness of the book from the tablet, something like that. But for the moment, it's sort of a giant experiment for the past 20 years, right? And there has not been this sort of evolution, cultural evolution, that tunes the writing system to our brains. I haven't spoken too much about that, but when I talk about this recycling, it's really a two-way process. Our brain is adapting to the cultural object, but our culture, the cultural invention of reading, has adapted to the brain. We've selected shapes that are super efficient to be processed by our brain, like T's and O's and so on. We've selected punctuation. We've invented spaces. Spaces were not always there. In the Roman times, there was no space, and now it created a lot of difficulty. And then inventing a little dot, which is not great, and then they invented the space. So all of that evolution has taken place with the actual book, and it has not yet fully taken place with tablets and so on, and I think we're still suffering a little bit from that.
I was actually going to ask the same question, but I'm now going to ask a more mechanistic question. When I get those emails, which have the first and last letter correct in every word, but all the middle ones jumbled,
right?
Which are very easy to read, I always think that I am able to read them partly because of the orthographics, but also partly because I'm continually predicting what's going to happen.
Yes.
And so are any of those predictions happening at the level of the visual input, or is it all at the level of the language? As in, is it really a word-by-word thing in the orthographic orthographic system, or is there a broader context like learning going on as well?
So I already spoke about the input system. There is all of these additional stages indeed of lexical access, of syntactic construction, and so on. And so as far as we know, they are all influenced by predictive coding. So that's a very important mechanism. I would need to look, but I think there is predictive coding at the level of the visual input, but I would need to look again to make sure of that. But surely that's a very important mechanism. And so in reading these emails where the letters are jumbled, first of all, the first and last letters are always preserved. That's very important. That makes sense with respect to what I showed. But also you're slower. And you're measurably slower as a function of how many transpositions have been put in. So it's partial preservation compensated by predictive coding and so on. And that explains why these dyslexic children, they complain, the letter position dyslexics, they complain, but it's hard to put your finger on a very specific deficit because they are slow and everything looks more ambiguous to them, basically. But eventually, in a normal text, they can get to the message. It's only when you have these lists that are carefully designed to have these special words, singe and cygne, that could be read in two different ways, that suddenly you realize that they have a major problem.
Okay. So I'd like to ask about evolution. Reading in evolutionary terms is incredibly recent.
Right.
Is it just a coincidence that there happened to be this part of the brain with a connection to the visual system and to the language system? Or do you think there might actually have been some natural selection in just the last few thousand years?
That's a great question. I deal a lot with it in this book, Reading in the Brain. I think no. For all the evidence, this is way too short time to have significant evolution of the genetics that create the scaffold the brain, especially if you consider that until recently, most people could not read. You speak of several thousands of years, but for all of this period, there were very few people that could read. And also, but that's more like a Woody Allen joke, but the more you read, the less you reproduce, basically. As well now. Okay. Several ways. But it's true. I mean, the more educated people reproduce less. We all know that. Okay, anyway, so... Joke aside, that means that we are recycling a system that did not evolve for reading. And that's why cultural evolution had to have a significant impact. We found the right shapes. It all started with shapes of objects that were close enough to the object so the visual system could still recognize them. That's hieroglyphs, for instance. And then somehow we found that we could simplify. We didn't have to make all of these very long drawings. Egyptian writing, was slow, and then they simplified it to just have these shapes that in fact are closer to the neural code that we use to recognize objects, line junctions, X's, O's, and so on. And we refined that system through cultural evolution until it fitted our brains really nicely. There's still a lot of learning to do, but it's relatively small. And the last point is, yes, we can show that in the young infant, there are already these connections from the ventral visual system to the language system. They pinpoint rather specifically the depths of the occipitotemporal sulcus where we see the visual word form area appearing. So the pre-existence of these connections probably explains why this area is always at the same location. I guess I didn't say this. It's in the same location in all of us within 5 millimeters or so, standard deviation. There is a gorgeous study by Sajin and collaborators at MIT showing that if you scan children at the age of 5, where they have not yet learned to read, you can predict where their visual word form area is going to be at the age of 8, just from the patterns of connections. So we really think it's the connections that create a very significant framework, basically, to constrain the system. And presumably, we could not read, we would not have invented reading if there was not this circuit. So behind that, there is a little philosophy of mine which says that cultural recycling is the key factor. We have a plastic brain, it's plastic enough, but it's not fully plastic. And the cultural forms that we see in other domains as well, like mathematics, symbols, and so on, they're all constrained by this existing brain architecture. Culture is not free, and we are not free of our biological architecture, basically.
Sorry, we have a question online.
Right.
So the question is, how does the dyslexic population experience, does it also experience difficulties in reading music? And maybe a question for me, is there a general relationship between reading words and reading music?
I don't think I know personally, and I'm not sure even in the literature we know so much about reading of music. We're just starting to do some brain imaging of that with my colleague Laurent Cohen. However, we know a lot about reading letters versus reading numbers, which is, I think, the same spirit of the question. And Nama Friedman has been showing that for every type of dyslexia, numbers are spared. It's really stunning. It's really like this very sharp division between the system for numbers and the system for letters. And it shows that there is really something very specific about the architecture for reading that I was describing. Music goes more with numbers, as far as we can see so far in our images. Music is a spatial language, and number and space belong together in the parietal lobe, and we see that the music system is recruiting areas that look suspiciously similar to the number system.
So on one hand, you showed that the brain under reading in like French readers, English readers, and Chinese readers look quite the same. And then you mentioned that like this kind of idea about the mechanism and the computation related to the order, the ordinal, basically the placement order of the letter is important in there, particularly presumably the word patch. But that's not the case to read Chinese, right? The order doesn't really matter in that symbolic reading. So what other computation is happening there?
That's a very good point. First of all, I want to show you one slide that I left out, because we did scan some English-Chinese bilinguals, so I can show you their brains. And you're completely right. There is a little bit of similarity, but if you are inside the same brain, so there is a big difference between group studies and single subject studies. In group studies, we find that Chinese reading is quite similar to alphabetic reading. But if you look at a single subject, you can find, in this case here, and you can find voxels that are just for Chinese, and voxels that are just for the alphabet to a lesser extent. There is still a lot of sharing. So all of the voxels in green are shared between Chinese and English here. But the voxels in yellow are just for Chinese. Okay. So there is some specific tuning to those shapes that are quite different from letters. And maybe you're right. It's not quite true, though, that the Chinese characters could be mixed up and you would still read. There is a position dependence. Some sub-characters can appear only at specific locations within. So I suspect some kind of generalization of the bigram coding scheme would work, space bigram, but you have to account for the 2D spatial position and not just the 1D spatial position, vertical as well as horizontal. But it's much more difficult to analyze, and we have not really analyzed it in this neural network. We should do that. Okay. Oh, and I forgot to say one thing. If you're Chinese, these voxels that like Chinese, they like faces. And we don't know why, but it's an empirical discovery. The competition between words and faces seems to be real, but sometimes we see a lot of overlap. And in the case of these Chinese voxels, it's quite clear. I mean, you can see the average here. These patches that have been selected because they respond only to Chinese, they also respond to faces. So there is lots we don't understand about competition versus collaboration. Maybe that's an important message. I insisted on competition on the cortex, but the advantage of reading is huge, and you refine your visual system by learning to read. It's not just that you don't destroy your face system, you shift it around a little bit, but you gain a lot in high resolution processing and so on. So the advantage of learning to read are huge.
Thank you, Stan. I think we're going to have to leave it there. Thank you for all the questions. I know a couple of you also had questions online, but we won't have time to discuss them now. Thank you also to our VSL interpreter who's taken extra beyond her time just to accommodate all the questions. Really appreciate that. And then thank you, Stan, for telling us how the brain reads and how we learn to read.
Thank you so much.
Thank you again. It really